Okay, so the next part is about the AB compartment. As we mentioned, when people look at the high-C map, loops are the dots in this high-C map. Domains are the triangles in this high-C map. And compartments, these are, if you zoom out even more, you can see here, these are the triangles, you see? But if you keep on zooming out, you will see that some regions are kind of whiter to each other and other regions are not so white. You can see here, like if you take the eigenvalue decomposition on the high-C map, you will see this kind of checkerboard. And this type of checkerboard, actually, once people look at the histone multiplication and the gene expression, what they realize is that the first eigenvector correlates with gene expression, histone marks, and many other things. And let's see, maybe the order is not as good. Yeah, so maybe I can show you here. What people found here is if you look at the chromatin that's packaged in the nucleus, this is the nuclear membrane, and on this there are many pores. You can see this basically nuclear membrane that has holes, and there's a nuclear envelope, and the holes allows, you know, when say some transcription factors or other stimulus, you know, kinase or whatever to get activated, they will go, you know, signals go in and out of the pore. And right on the outside, these are more, you can see compartment B. And in this region, you see mostly closed chromatin. If you do DNA methylation, if you do K27 trimethylation or K9 trimethylation, they are more enriched. So these are the kind of the red regions in here. These are kind of more closed chromatin. Interestingly, they also have lower transcription. Usually the genes around there are not activated. And also in DNA replication during cell cycle, they are the last region to be replicated. Whereas the blue regions in here, these kind of are the transcriptional factory of the nucleus. They have more open chromatin, they have higher transcription, they are just kind of active. And in DNA replication, they are replicated earlier. And that's kind of translated into this type of checkerboard. You can see within each chromosome, there are regions that are more active and there are regions that are more repressed. And you can see this much, much bigger chunks, I would say probably in the order of hundreds of KB or even megabase resolution. And then when people are looking at it in the overall contact map, you can see here that this is the interaction map. So overall, we can see when things are close by, they are more likely to interact. But when they are further away, this is really looking at, you know, like 1 megabase, 10 megabase, or even 100 megabase. You can see definitely as you go further or further away, they are, you know, they are just really far away. They are unlikely to interact with each other. And so if you look at the contact probability map, you do see this type of interaction decay with distance. But people are wondering what type of, you know, how fast does this decay happen? So in chromatin interaction, there are two possibilities or, you know, two potential models. One is called equilibrium globule, which means it's called random. Even if it's random, things that are far away are still less likely to interact. And you can see here, when you look at this, it's kind of random. And if you do a cross-section of this ball, you know, there's a nucleus ball, you can see it's kind of all over the place. Whereas the alternative model is called a fractal globule. This is how ramen noodles are made. So in some sense, the things that are close by, you want them to still be closer to each other to form slightly better organization. And so for that, if you use you know, like you cut the cross section of this nuclear ball, you do see that you know, the blue regions are, and the, you know, the red regions are roughly close to each other. And so people did simulations to see if you have equilibrium or fractal, how does that influence chromatin interaction? And this is based on the simulation data in here. And you can see if it's, what you will see is that when things are really close, it doesn't really matter whether it's fractal or equilibrium. In fact, when we are really talking about this short distance, we're still at the loop and TAD domain level. But when you are looking at further away, the compartment level, you can see the equilibrium globule is not as good. They don't have, so when things are a little further away, it's a lot harder to interact with each other. Whereas if you have a fractal kind of globule, the things that are further away, they can still be close to each other on the chromatin or the overall nuclear packaging. They're closer to each other and are more likely to interact with each other. But when they are, of course, really, really far away, then you don't really care. They are just too far to interact. And so based on the simulation and the actually observed high C, people believe that the chromatin are more packaged like a fractal globule instead of a complete random equilibrium globule. This would ensure that things that, basically within the chromosome, things that are, you want to have the things that are closer by to each other in genomic distance to still try to stay as a big chunk together in the 3D structure. This is actually helpful because you can imagine when the chromosome is being replicated, or you want to more chromatin interactions, this ensures that things that are close by are still together. They are kind of more organized. Also, you can imagine, as we mentioned, we're trying to package 44 miles of yarn into a basketball. If you don't have the correct packaging, at the end, you could have a loop that gets stuck in the other loop. You can imagine if you have a long yarn, you try to open it up, you will be crazy, right? And so forming these type of fractal globule, make sure that things are still roughly organized in chunks. If you have to amplify something, if you have to express something, some regions can move in or out of the nuclear center from the compartment A to or B. They can still do this in a coherent fashion, which will make gene regulation more coordinated and less likely to entangle each other. And I see some questions. What each region is so this is just, sorry. This is just an example. You can imagine, for example, orange is chromosome 1 and red is chromosome 2. And even though they are still, some of them in the outside of the nucleus, someone in the inside of the nucleus, still they roughly try to stay together rather than completely free floating in the nucleus. Does that make sense? Let me look at the chat. Sorry, I'm losing my chat window. Oh, there. How do we define when we go from blue to green? Oh yeah, this is just an example. People are using this to do simulations to see whether close by regions are still close to each other. You can do this with the chunks of a region. Can you clarify why probability still go down as distance increase in the equilibrium? So even if you have free yarns, and when things are close by, they're still stretching each other, but when they are really far away, they don't have relationship to each other anymore. So they are free to float anywhere. But if they're close by, they're physically linked. So if one moves, the other one does get kind of dragged a little bit. Does that make sense? So the distance still matters. Let's see, we have one more question. Can you clarify, sorry. I think, do we have more questions? Can you clarify why probability still goes down as distance increase in the equilibrium? Yeah, I think that's the question we answered already. And so yeah, by this time, what people can already see is that there are chromosome territories to ensure that things that are physically close to each other, or even genomically, if they are close together, you still try to organize them in a region in the nucleus rather than having them completely free floating everywhere, just to make sure that the chromatin doesn't tangle each other when you have to have coordinated activation or repression of each other. At the same time, when you look at how they are packaged within the nucleus, there are compartment B regions, which are in the center of the nucleus and also on the kind of the boundary or the nuclear envelope. These regions are usually more closed on chromatin. They have lower gene expression, whereas the blue region are the more active open chromatin region, have more gene expression, transcription factor binding, and enrichment in there. Interestingly, because of these nuclear pores, so imagine you have a very important transcription factor that suddenly get activated, and it's a very important factor. It can actually enter into the nuclear pore and go to this red compartment B region, and with enough binding to the chromatin, they can drag a particular region out from the red to the blue, and then that region can get activated. And so you can have some of these red regions are constitutive, but there are also many regions that are cell type specific, depending on what transcription factors can go to the nucleus, where they bind. They can take some of the regions from a compartment B to a compartment A to start transcription. So you can see, in general, chromatin is very, very important for how we understand genes are regulated. And so in the 4D nucleome, people are still also looking at high C during cell cycle, during G1, G2, S phase or metaphase. During cell cycle, what happens? And you can see during the G1, G2, or even S phase, you can see a compartment and also the TAD domains. Whereas in the metaphase during cell cycle, I guess the cell is dividing into the really dividing into two different cells, and the chromatin is really highly, highly compacted. You actually lose the TAD domain, and you also lose the compartment, because basically at this level, there's no transcription. You don't really need the interaction. During metaphase, genes are not transcribed, and also chromatin is highly compact. I guess everybody is in the B compartment. Therefore, you kind of lose that information. But then as the cell is again dividing into another new cell cycle, when the chromatin start to loosen up, the motors start to go in, they will form the TAD domain again. They will also form the compartments again. Okay, so that's a kind of a dynamic situation. Sorry, this part. Yeah, and also interestingly, what they observe is during the cell cycle, during the interface of cell cycle, you can see the cell is changing between the equilibrium and fractal, depending on the regions. And so during the cell cycle, the contact map also can change. You can see here. Yeah, so this is the fractal interaction. This is the M phase during the cell cycle metaphase. This is the purely equilibrium. So it's kind of in between the fractal and the equilibrium. So you lose a little bit of you lose the TAD, you do lose the compartment, but the cells still try to organize the chromatin a little bit in the M phase. And so it's not completely equilibrium. It's kind of in between the two. Okay? Yeah, so you can imagine chromatin is being controlled by the transcriptional activity, but it's kind of a chicken and egg, right? So during cell cycle, when the chromatin is super compact, there's no transcription. But after the cell cycle, the transcription factor will start to bind, the motor will go in, it will start to form those TAD again, and kind of segregate into the compartment A and compartment B, but that will also influence which genes get turned on, which genes are turned off. but that can also be determined by what transcription factors are available in that cell. And so all these interactions with, how proteins are interacting with protein, proteins are interacting with chromatin, and chromatin are interacting with chromatin. All these mechanical and physical binding and interactions controls what we see in the Hi-C experiment. Okay, questions?